Hello, and welcome to the Onion IQ podcast. This is Alice Formiga of eOrganic, and our guest today is Dr. Mussie Habteselassie of the University of Georgia. He's going to be giving us a brief overview about the soil microbiome in organic onions. He's researching this topic as part of the USDA NIFA-OREI funded project Onion IQ, improving quality, resilient soil health, and food safety in organic onion in the southeastern and southwestern United States, led by his colleague Dr. Bhavesh Dutta. So today I'd like to welcome Dr. Habteselassie, who is a professor of soil microbiology at the University of Georgia Griffin campus. His research focuses on the impact of agricultural management practices of soil microorganisms. He's also interested in the fate of pathogens in soil and their transmission to fresh produce. He's going to give a short presentation, and then I'm going to ask him some questions about how soil microbiomes can help suppress onion diseases on organic farms and what farming practices can encourage this. So with that, I'm going to pass things over to you, Dr. Habteselassie.
Thank you very much for the kind introduction, Dr. Formiga. I will briefly give an overview of soil microbiome, and then with Dr. Formiga's question, we'll get into some specifics about the project, the IQ project. So to start with, I'm going to define what the soil microbiome is. I will start with microbiome. The word microbiome is a combination of two words. Micro is for microorganisms. Those are organisms that we cannot see with our naked eyes. We need some kind of assistance to see them because they are so small. And then biome means a collection of microorganisms under a certain climate conditions in a certain geographic region. So when we say soil microbiome, we are talking about a collection of these tiny, small microorganisms that we cannot see that live in a certain specific soil that's found in some area under a certain climatic condition. So what does this microbiome include in a soil system? We have a range of microorganisms that are included in the microbiome that they live in the soil, and they have been listed in this table here. These include archaea, which is one of the biggest group of organisms in soil and the smallest, and all the way to protozoa and nematodes. As you go from archaea down to protozoa and nematodes, their sizes increase. So nematodes and protozoans are bigger than archaea and bacteria. And then I gave a genus example, an example of each group under the second column, and then their sizes. So for example, archaea is the smallest, and its size is in the micrometer range, which means, well, micrometer is the millions of a meter, so it's very, very small that we cannot see that organism with our naked eyes. And then when you look at abundance, how many cells of archaea you find, if you just take a gram of soil, it can range anywhere from 1 billion cells to 10 billion cells. So you have humongous abundance of these organisms in the soil. So you can do the same thing for bacteria, fungi, algae, protozoa, nematodes. So collectively, all these group of organisms that live in soil in a certain area under certain climatic conditions, they are called the soil microbiome. So why are they important in soil system, in our environment? Because they provide a lot of important functions that enable crops to grow, that support crop growth. We call these services provisions. One of the most important services that microorganisms provide, soil microbiome provide, to support crop growth is what we call mineralization, especially in an organic system where we have a lot of organic inputs. The nutrients that are occupied or contained in the organic amendments have to be mineralized or changed into inorganic forms or minerals. For example, in this case, the organic nitrogen has to be changed to ammonium or nitrate for it to be used by microorganisms. So this conversion from organic to inorganic forms is carried out by microorganisms. This soil microbiome we talked about. Without them, the nutrients in the organic amendments are not going to be available to the crops. So they play a very important role in making nutrients available in forms that can be used by crops. Another important function would be nitrogen fixation. Basically, they make the nitrogen form, which is most abundant in the atmosphere, which cannot be used by crops, into a form that can be used by plants or crops, which is ammonium. But there are other free-living microorganisms that mediate that catalyze this process to make nitrogen more available for crops. Another important relation between microorganisms and crops is what we call the mycorrhizal relationship between fungi and crops. Is a very important relationship that enables the accumulation of phosphorus and water for crops from areas where their roots cannot reach. So these fungi produce a structure called hyphae, or it's also called mycelium or mycelium. So this structure is hollow inside, so it becomes an extension of the root, and it can explore the part of the soil that cannot be explored by the root because the roots don't grow that far. So they funnel back phosphorus and water into the root system, and then the crop responds in kind by giving them what we call exudates, these organic molecules that the fungi or the fungi can use as a source of energy. Another important function that soil microbiome provides to crop growth is disease suppression. Soil microorganisms can help fight diseases by these three mechanisms. A very active, a very diverse soil microbial community poses a very intense competition to the pathogens, thereby suppressing their spread. There are also a lot of microorganisms in the soil that produce antibiotics. A lot of streptomyces are known for producing antibiotics. We have a lot of Pseudomonas and Bacillus species that are known for producing antibiotics that can fight bacterial pathogens in the soil, thereby protecting the crop from diseases. And then we have a little bit bigger group of microorganisms, such as nematodes and protozoans that we talked about, that directly consume these pathogens and prevent them from causing disease. For example, this figure that you see on the left side, on my left side, you see an image of fungi eating protozoans. And then on the right, you have bacteria eating nematodes. So they help these protozoans, and nematodes help prevent or control the spread and the growth of pathogens, which is a very important function, a very important service that they provide to crops. I just want to show this figure to illustrate the fact that when you have a diverse soil system that has a very diverse microbial community, here you see there are, in this pie chart, you have a lot of divisions, a lot of pieces, okay, as compared to the one on the left side here. So here on the left side, the one I'm showing you, you have low microbial diversity. On the right side, you have high microbial diversity, as you can see, because they have more pieces. That means you have more robust, very diverse group of microorganisms in the soil versus this one. So when you have low diversity of microorganisms in the soil becomes conducive to diseases. It doesn't have as much power to fight diseases through the mechanisms that we mentioned earlier in the other slide. But when you have a soil that has a very diverse microbial community, as you can see on the right, you don't have as much disease severity, as you can see. So this figure on the y-axis is the disease severity. On the x-axis is the host plant cultivation time, growth time. So you see a very severe disease outbreak in the soil where you have low microbial diversity as compared to the soil where you have very high microbial diversity. So very active, very diverse microbial community or soil microbiome is very effective in combating diseases. So soil microbiome is a very important component of soil health, which is defined by USDA as a continued capacity of the soil to function as a vital living ecosystems to support plant and animal and humans. So the way the soil supports plants, animals, humans is what I mentioned, some of the the services that I mentioned earlier. And USDA has these 4 soil management principles that promote soil health, and one of which is to maximize biodiversity. So it's really key that we have very diverse, very robust soil microbial community in the soil system to improve soil health. Thereby, the soil will be able to provide a lot of services, supports to the plant or the crops that we grow. So you can see here's an electron microscope image of a seedling growth, which is surrounded by the soil. And you have a lot of microorganisms here that are active that can provide some of the functions that we mentioned earlier. So whatever management practice we employ, especially in organic systems, we want them to promote soil health. We want them to improve soil health, as you can see in this dotted green line, or at least maintain the soil health, as you can see on the flat line, the solid line. We really don't want them to decrease or negatively impact the soil or degrade the soil health, because if we degrade the soil health, then we need to compensate for the lost provision that would have come from the soil microorganisms by using external input. So if you have a system that's improving soil health, you don't need as much external input to make the system productive. So because the crops already have a very robust support system in the soil through the action of the microorganisms that we already mentioned and discussed. So finally, I just want to briefly mention the different ways that we measure soil, we call it soil biological health, but what we are measuring is how healthy the soil microbial community is in the soil system. So we can measure indicators that will tell us how healthy the soil microbial community is. Okay, for example, by measuring respiration, we are indirectly measuring the decomposition of organic matter, which we talked about earlier as being very important in releasing nutrients, right? So we can measure that process and see how healthy the soil microbial community is. It tells you the activity of the microorganisms. We can measure the activity of enzymes that are produced by microorganisms because they produce the enzymes because they use them to catalyze or mediate processes that we already looked at. For example, nitrogen fixation, for example, nutrient cycling. So if we have very active and robust microbial community, we will have very high and robust enzyme activity as well. In this case, I gave you an example where urea in the soil system, which can be applied through organic amendments, is not really, the nitrogen in urea is not readily available to crops for use. It has to be first hydrolyzed to ammonium. So this hydrolysis of urea is catalyzed by an enzyme called urease, which is produced by microorganisms. So if you measure the activity of urease, it will tell you directly the activity of the microorganisms that make the nitrogen in urea available for crops. So that way you can measure how active the microbial community is. The same with, I talked about how a very diverse, active, and robust microbial community is important for disease suppression, for example. You can measure that diversity by looking at the abundance and composition of some key group of organisms that are involved in key functions, such as nitrogen fixation or mycorrhizal relationship. So you can measure the abundance of nitrogen fixers, mycorrhizal fungi, and so forth. And now, because of the advancement in technology, we can actually sequence their DNA and identify these different groups and how they change in response to different agriculture and management systems. Now I'll just stop here and allow Dr. Formiga to continue with the Q&A.
Okay, well, that was really interesting that there's so much activity going on in the soil, and there's such a variety of different creatures, some of which I'd never even heard of, that live in there and how small they are. So it really must be fascinating to be able to study this with all the advanced tools that we have and things like electron microscopes. But in terms of how what you're doing specifically addresses organic onion diseases is probably the most relevant thing to this project. So onions are known to have relatively small root systems. So how does that affect their dependence on microbes?
It's a great question because of that property, they won't be able to explore much of the soil system. So where the microbes come in is by, they form relationship with the onion. For example, it's mycorrhizal relationship. Onions are known to create mycorrhizal relationship with fungi. Depending on the type, the variety of fungal communities you have, they become an extension of the root system. And so it's basically, they look like, they are thread-like structure. They're hollow inside. So they attach themselves to the root and then they grow away from the root looking for nutrients, mainly phosphorus and water. They can actually grow farther, way farther away from the root and explore parts of the soil where the root cannot reach. And then once they do that, because they have this tubular thread-like structure, they can import back the phosphorus and the water to the root and help the plant with acquiring phosphorus and water. And then the plant will actually respond in kind by providing them with this organic molecules that they can use as a source of energy through the process of oxidation.
So what are the names of these microbial groups that are so important for onion?
So they are found in they are fungi, but specifically they are found in the phylum Glomeromycota and the genus Glomus. So we have a variety of them. Like you have a huge abundance of these fungal groups that do that.
Ah. So what farming practices most strongly can encourage these?
So for especially these fungal groups that form mycorrhizal relationship, they're very sensitive to disturbance because the structure that I told you about there, that's just basically thread-like structure. So if there is any kind of tillage disturbance, any kind of plowing disturbance, it's going to break down these structures. So that they are very sensitive to any kind of disturbance in the soil system. They also thrive through, if you have diverse set of plants or crops, if you do crop rotation, is very important because that helps with pathogen control. As well, in organic amendments is also helpful in terms of maintaining these fungal groups because fungi are heterotrophs. Heterotrophs mean they depend on organic carbon and nitrogen source. They don't make their own. They don't fix carbon or they don't fix nitrogen from the atmosphere. So they depend on pre-made organic molecules that can be provided by organic amendments.
Okay, so this project is mostly about Burkholderia species in onions. So how do these microbes help prevent or control Burkholderia?
So as broadly in general, not only Burkholderia pathogens, but other pathogens, if you have these very diverse and active microorganisms, they prevent them from spreading through by posing a very strong competition for nutrients and water. And some microbial communities, as I mentioned, which are found, for example, Pseudomonas Bacillus subtilis, Pseudomonas fluorescens, they produce antibiotics that can control the spread of these Burkholderia pathogens. So these are some of the mechanisms, but they also have nematodes and protozoans that can feed on these pathogens.
Can one introduce these into the field somehow?
Yeah, there are a lot of products, they are called biological products that have this, especially Bacillus subtilis and Pseudomonas fluorescens in large abundance. They grow them in the lab and they can be applied to the land. There are products that have these microorganisms. They call them sometimes biological products, sometimes biostimulants, but there are some biological products that have these microorganisms.
Wow. Okay, so what have you found in the first 2 years of your trials for this project?
So the objective for the first 2 years of the trial was to
Well,
evaluate.
basically, what have you found so far? Is pretty
Yeah.
much. Yeah.
So I just want to mention first what we are doing, and then I can tell you
Okay.
what we found out.
Yeah.
So the objective was to evaluate two cultural practices, biofumigation and solarization, as ways of controlling the Burkholderia pathogens in onion systems. Because in organic systems, since we cannot use chemicals, we have to look for other cultural practices such as solarization and biofumigation. And what my role in this project was to see how these practices, cultural practices, impact the total Burkholderia population in the soil. So both the pathogenic and then pathogenic. By the way, there are some beneficial Burkholderia species as well, and in addition to these pathogens that we are really interested in, focused on. So that's what we did for the last 2 seasons. So for the first year, the second year study is still going on, but for the first year study, so we have seen some changes in Burkholderia populations, including the pathogens, and there seems to be some slight impact of this cultural management practice on the Burkholderia species. But it depends on when you take the samples. Like we have the pre samples during the transplant time, and then the post-harvest time. So in the middle of the season, we see some impact of these cultural practices on the Burkholderia population. But for the second year, we just collected the last batch of samples a few weeks back, so we are still analyzing the samples and looking at the data. what we are going to do is there is another study as well, which is looking at the use of organic amendments, poultry litter, and commercial organic fertilizer, how that impacts the fertility of the onion system. And then in the last 2 years, 3 and 4, we're going to combine these cultural practices with the fertility practices and see how it impacts the soil microbiome in general.
Great. Okay, two more questions. So are there any challenges that you faced during this study? And if yes, how did you approach them?
I think so far so good. The project has been going smoothly, knock on wood. So we hope for that to continue throughout the study.
Sounds good. Okay, and I guess since we're coming to the end, what is one takeaway that you want growers or listeners to remember?
I think this is just for pretty much every grower. If you nurture the soil, the soil will nurture the microorganisms, and the microorganisms are going to take care of the crops. So if the soil is properly taken care of, you don't have to use a lot of external inputs, and you'll have a very robust and productive system, is what I would like to say.
Okay, thank you so much, Dr. Habteselassie, for sharing your presentation today. And we will look forward to hearing more as the project progresses.